Good evening, everyone, and welcome to the last Science Cafe of our Ocean Science series this fall. We're glad that Magpies was able to bring us inside again. Last month it was raining, and this evening it's pretty darn chilly out there, especially for us Tucsonans who aren't used to the cold weather. So it's exciting. We're glad that we're warm inside. Welcome. As many of you already know, if you've been here before, my name is Shepard Reed, and I work at the Glendora Science Center and Planetarium. We have great planetarium shows and exhibits that are great to bring visiting family to over the holidays. So if you have family coming to town and you're wondering, what do we do? Come and check out the Science Center Planetarium. Learn some science, see a great show. We have our outreach rack back there. It says Explore More Science across the top. And there are lots of great science things to do in and around Tucson. So check that out. We have a Arizona Science Adventure Map brochure. And I know a lot of you are regulars, so you're quite familiar with all the amazing science here in the Tucson region. And with that, I am very pleased to introduce Katie Morgan, the manager of marine ecosystems at Biosphere 2, and she is going to tell us about the ocean at Biosphere 2. Some of you may have visited there before. It's exciting, the research that's going on now, and so please give a warm welcome to Katie Morgan.
All right, well, hello everybody. Thanks for coming out tonight. I promise this makes me feel like I'm doing a concert, but I won't sing for you. I promise you that. So, like Shepard said, my name is Katie Morgan. I work up at Biosphere 2. I am manager of marine systems. How many of you have been to Biosphere 2 before? Heck yeah. How many of you have been in the last 2 years? Awesome. Okay. So we have lots of exciting research going on at Biosphere 2 right now. Arguably, I think some of the most exciting things to ever happen inside of the facility are happening right now today. If you haven't been to Biosphere 2 before, my clicker is on, that will help. Biosphere 2 is a earth science research laboratory about 45 minutes north of here. It is owned and operated by the University of Arizona. And inside of this facility, we have a tropical rainforest, a 700,000-gallon saltwater ocean, which is what I'll be talking to you about tonight. We also have a marsh ecosystem, a savanna, a desert, and about 2 acres of technology underneath all that biology that runs it all. So it's an incredible facility, and we like to compare it as a tool to the earth sciences, just like the Hadron Collider might be a tool to physicists, or these massive telescopes that might be a tool to astronomers. We use Biosphere 2 to understand our planet, which is very important. We need to know more about our planet, so we're working very hard to not only understand our planet, but understand the things that our planet is up against and the solutions that we can come up with in order to combat some of those things that our planet is up against. The reason that Biosphere 2 is such a great place to do that research is because we are very unique. When you think about doing research, a lot of times you think about a laboratory. You might think about scientists in lab coats in a small building, in a basement, that you're not really seeing what they're doing. And that's a really important part of science. You need that laboratory science. But if you're doing laboratory science that needs to then advance to a field setting, it's really hard to go from lab to field. It's a huge jump. So what Biosphere 2 offers is the scale and the control to do these massive experiments to fully understand the science on an ecosystem level, which is really important for understanding our planet and understanding how this science impacts our planet. Now, when we think about how that's important for the ocean, scale is definitely a big thing for the ocean. So our ocean at Biosphere 2 is actually the world's largest ocean research tank. So there are larger aquarium tanks, but these aquariums maybe see, millions of people every year. They're not going to let you touch that tank. They're not going to let you manipulate the variables or stress out their animals. They want it to be crystal clear for their visitors. We want to stress out our animals. We want to do science in here, and we want to show the public what that means. So we have this really valuable tool at our fingertips to do some really cutting-edge science that I will tell you about today. So, when you look at this photo, what is the majority color that you see? Blue. Yeah. That's because about 72% of our planet is ocean. So it probably is a bit important, right? So our oceans drive a lot of things on our planet, and they're incredibly important for a lot of reasons, which I'm sure you know about. And I'm sure you know about this too, is that our oceans are under some immense amount of pressure right now. There are a lot of things these oceans are up against, and this will be my small little smidge of doom and gloom, and I'll bring it back, I promise. I'm going to bum you out for a second. So one of the things that our oceans are really up against right now is temperature. So temperature is increasing in our oceans, and what is happening is we've seen this happen forever. Since the oceans have been around, you've seen fluctuations in temperature. What's different today is that our oceans are staying warmer for longer, and that is really affecting our systems. The other thing that we are seeing increase in our ocean systems is when we see an increase of CO2 in our atmosphere. The oceans are really great at taking things up. They suck up that heat. They also suck up that excess CO2 in the atmosphere. That's great for us as humans, so we don't have all this excess CO2 and heat in the atmosphere. However, we do have acidic oceans because of this. When they take up that CO2 from the atmosphere, a chemical reaction occurs that actually makes the ocean more acidic. The other thing that the oceans and the animals are up against in the oceans is fishing. So fishing is becoming more sustainable now, but we could always be better. Sustainable fishing involves, making sure that you know where your fish are being caught, how they're being caught. These massive fishing vessels that go out with these large nets a lot of times aren't thinking about the other animals that they're catching along with that species that they're out to catch, the target species. And the species that they get on top of their target species are not being collected. They're being thrown back in the ocean, but they are no longer alive. They're most of the time pretty beaten up by the time they get out of those nets. And arguably one of the most talked about things in the media right now is plastics. Plastics is a massive problem in the ocean. This is a real-life picture of a guy surfing off the coast of Bali. This is a reality of our oceans around the world, unfortunately, that we are seeing more and more pollution occur. This pollution occurs in a couple different forms. So we have what you might consider our macroplastics. These are the larger plastics, the water bottles, the plastic bags, the soda can. So these are the things that we kind of think about and we're trying to reduce. Something that we might not think about are the things called microplastics. So these are these small little beads of plastic that are hidden in a lot of our cosmetic products and our toothpaste and our body wash as an exfoliator that then makes it down the drain into the oceans, into our fish. And we find about 30% of the fish in farmers markets or fisher markets have this microplastic in their flesh. So by us using it, it's coming back around to us. So that should make us care, right? All right, so how does this relate to the Biosphere 2 ocean? The Biosphere 2 ocean has been at the forefront of understanding some of these problems that the ocean is up against since 1996. So Biosphere 2 was established in 91, and they ran some sealed missions inside. And then in 96, Columbia University actually took over. And Columbia was really interested in CO2. So we had seen CO2 going up in our atmosphere, but we weren't really sure how that was affecting our natural systems. So we knew it was going up, but how did that affect our rainforests? How did that affect our oceans? Is there a tolerance level? Is there a level that the trees say, I'm done, I'm not taking up any more CO2? Is there a tolerance level for our corals as they become a more acidic environment? So they were able to use the Biosphere 2 ocean because the original experiment actually brought in corals from the Yucatan Peninsula. So there was a very diverse coral reef inside of the Biosphere 2 ocean. These are the best pictures we have from '91, so that's all I get. But this system was a really important place to do some of this early research. So they were actually able to acidify this water. And they were able to look at future climate conditions. When the CO2 continues to go up in the atmosphere, how does that affect these coral reefs? And corals are really unique animals. Brought skeleton for you. So they are animals. They actually build this skeleton out of calcium carbonate. So they pull ions available in the water column to build this skeleton. And when those ions become less available because of acidification, they have a really hard time growing this skeleton. So they get pretty stressed out. And when they get stressed out, they have this little, which is a fun word, it's called a zooxanthellae. It's a plant cell that actually lives in the tissue of the corals. And when the coral gets stressed out, that plant cell actually gets expelled from the coral. So it says, okay, I'm done, goodbye. But this is an issue, because that plant cell actually provides about 90% of the energy for corals. So now they're not only stressed out because they're living in acidic water and they can't build a skeleton anymore, but now they don't have the thing living in their tissue that provides them their energy. So now they're having to filter feed for food. So they are at the tipping point. And you might see how these corals respond to this. This is called coral bleaching. So they turn this stark white because when that plant cell leaves, that zooxanthellae leaves, it loses coloration. This stress response was actually shown in the Biosphere 2 ocean. So they were able to directly relate the increase of CO2 to bleaching. That was pretty important. That's a study that we have used for a long time now, taught for a long time now, and is kind of the forefront of starting coral reef research at Biosphere 2. So when you see a reef like this, you start to see things like this. This is maybe a reef that you would see that's a little bit on the healthier side. But unfortunately, what we're seeing happen pretty rapidly today is reefs looking like this. This is a pretty big problem because coral reefs play a very important role in our oceans. A lot of times we actually compare coral reefs to big cities because they have all kinds of structures and functions and diversity. So swimming in a coral reef is similar to walking down a street in New York City. You see all kinds. So there's a lot of comparisons with these massive cities to these coral reefs. And they're also pretty important for us. So they provide a place for about 25% of marine biodiversity. So about 25% of the ocean spends some point in their life on coral reefs. It's pretty impressive when you think about how much surface area corals cover around the world. I think it's like 0.01% surface area on our planet coral reefs cover, but 25% of marine biodiversity lives there at some point in its lifetime. So pretty important for that. They're also really important for fisheries. So how many in here like to eat fish? Okay, so coral reefs are important for you to continue to have fish on your plates. But also for communities that are coastal, that fish is really the only protein that they have. Corals are very important for that. The world's economy depends on corals, so about $36 billion of revenue is generated every year that can be directly related to coral reefs. So whether people are going on vacation to go scuba diving or just to go to a pretty beach, that revenue can be related to a coral reef. And then they also protect our coastlines. So this is a kind of a barrier around our land that when there's massive storms or just really traditional waves, it protects our land from erosion. So they are important for us as humans too. When we live on these lands, these corals are essential for us to continue to live in certain places around the world. Unfortunately, they're under some pretty major stressors right now. So CO2 is no longer the main stressor to corals. So we've lost about 50% of the Great Barrier Reef in the last 30 years. So in the last 3 decades. That's fast. And that loss can be pretty much directly contributed to temperature. So temperature, the increase in temperature, is the main threat to coral reefs today. So the Biosphere 2 ocean, to bring it back, has been an important place for this research for a long time. But after Columbia University left, the ocean wasn't doing so well. So most of the corals had died because of that CO2 experiment. And if they hadn't, there was really no one taking care of the ocean after the fact. So we do not, at this moment, have any living corals in the Biosphere 2 ocean. We have some very charismatic small organisms. This is just an example of one. But we really have a lot of algae. So this is pretty typical. When we see coral reefs die off, we see this kind of degradation happen. We see mass algae blooms, they overtake the reefs. If there's no kind of reason for animals to be there anymore, the animals leave, and algae just really takes over. So this is kind of the state, I have a little bit of an update, but this is kind of the state the ocean is in today. But we're working on changing that. So we have a really exciting research project happening right now where we are going to keep putting corals back into the Biosphere 2 ocean to look at the main stressor to corals today, which is temperature. This is a project that I am not alone in. We have a massive team working on this. So we have our Biosphere 2 ocean team, which is staff that works for the university that is involved in this. We also have a scientific advisory board. So these are the people that are the best of the best in this field, doing work in their laboratories. And then about 40 international partners that are also doing this work, that see value in bringing their science to Biosphere 2. Now, why is this so unique? Like I mentioned in the beginning, it's all about scaling. So there's amazing work being done in laboratories where they're looking at these solutions to saving coral reefs. What are the things that we can do to make corals more resistant to temperature and more resilient in the future? They are finding amazing things, but what we're not able to do yet is scale that up to nature. Now that's fair, because we can't get permits to do something if we don't know how it's affecting the full ecosystem. That's where Biosphere 2 comes in, is that we can understand how these solutions are affecting a full ecosystem. So in order to get to corals, we had to do a little work. We have been in the process of going from slime to corals. So there's a very famous paper called The Slippery Slope to Slime, which is talking about the degradation of reefs from healthy reef to an algae-covered degraded reef. We're going to do the opposite. We're going to go from a degraded reef back to a healthy reef and track it. So this has really been the last year of our lives, is undergoing this monitoring, intensive monitoring, doing multiple samples, understanding what it means to have a degraded reef, and then tracking it as we bring it back to a healthy reef. And in order to do the research that we need to do, we've also had to add some life support. So what you're looking at here is a titanium-plated heat exchanger that allows us to manipulate the ocean temperature to 700,000 gallons of water, 2 degrees Celsius in a span of 24 hours. Right? That's crazy. It's some intense control, and it allows us the opportunity to really understand the stressors that these corals are under. This is realistic. This is the kind of temperature swings these corals are facing in the natural systems. And this is hot off the press. We're really excited about this. We actually just started using this heat exchanger about a month and a half ago. So it's been a process, and I just want to show you. So this is from November 19th. We actually warmed the ocean. This is us testing it out. So we warmed it 2 degrees Celsius, 24 hours. It worked like a charm. Then we stabilized it for a little while, brought it back down, and then brought it back up. So we are stabilized now at about 24 Celsius. That will be what we're at until we start adding corals to the system. So that was a pretty satisfying thing for us to watch, to see that we can truly manipulate this ocean temperature. And for me, it was pretty exciting because now our ocean is 24 degrees Celsius, which is a much more comfortable temperature to be in the water at, versus if we just let it do its natural thing on a night like this, it might be like 18 Celsius. So it makes a much happier diver. The other thing when it comes to control is thinking about what these corals need. So not only do we need to be able to manipulate the temperature, but we need to have a lighting structure. So what this is right here is a drawing of the lighting structure that we plan to put in this ocean in the next year. This is a big lighting structure. It has room for about 288 halogen and LED bulbs. So this allows us to have the proper amount of light on these corals, because like I mentioned, they have that plant cell that lives inside. So having light is pretty important. And they didn't have that in the initial experiment, and these corals really showed stress because of that. So we want our corals to only be stressed out when we tell them to be stressed out. So we need to make sure we have enough light for them. When it comes to doing experiments at a larger scale, we have to kind of start small sometimes. So we've got a 700-gallon raceway, so we call this our coral raceway. We will have 5 of these, and this is where we will start experiments and run experiments at the same time as we're doing things in the ocean. So we will start our corals in this tank in the next couple of months here. And if you've ever been to Biosphere 2, you can see it's a little bit of a complicated system. So getting big pieces of equipment like this up into small rooms takes some help. So this is Jimmy, one of our maintenance guys, that actually had to solder off the door in order for us to get that tank up into that room. So it's definitely a team effort to understand coral reefs. We need a little bit of everybody. All right, so when we talk about understanding reefs, or understanding the Biosphere 2 ocean, that comes in many ways. So we have been tracking everything suspended in our water, collecting data every 15 minutes. We've also been taking samples once a week for nutrient data, twice a week for their microbiome, and observing the macrobiome as well. So everything from algae to invertebrates to fish. We've been fully understanding this as well. We've partnered with a lab on campus with Jeremiah Hackett, who's understanding the microbial dynamics going on in the system. This is essential for us to understand because before we put anything new in, we need to understand what we have so that we can watch it change. Because inevitably this is going to change. The microbes are going to shift the minute we put something new in. That's going to change depending on what we have. So Jeremiah is an essential part of this. We actually have some of his undergrads working with us. So we try to get as many undergrads involved in this project as we can. There's a lot of people interested in marine science in Arizona. So we try to engage them in the only ocean in Arizona. So this is what some of those baseline surveys look like. Some of our algae. So we see things that you'd see in the natural system. If you were to go out to San Diego with a microscope, you would see the same kind of things. These dinoflagellates and diatoms. So we're just kind of fully understanding that. So we're not only just understanding the water column. Biosphere 2 ocean has about 30 years of history of sediment. And so we are also taking sediment cores in the Biosphere 2 ocean and understanding the dynamics and the changes that we've been able to see over time using the sediment. So Jake, who is he was an RU student with us this summer. He's from Xavier University and actually lived out of Biosphere 2 for 3 months this summer and did this research with us. He just presented at a big conference last week, so we are very proud to have him on our team. So while we're understanding all those different dynamics, we are also tackling this algae. So this is no small feat. If you think about 700,000 gallons of water covered in algae, it takes some help. So we've introduced some key invertebrates and vertebrates, so fish and other animals to help us eat this algae. We've also removed a heck of a lot of it by hand, and I will show you that in a minute. But we've also added, this summer we added we had a blackout period. So we put a shade cloth over this entire ocean to black it out to reduce some of the algae spores in the system. These are some of our charismatic organisms that we have living in our ocean today. So pencil urchin, these are my favorite guys, these emerald crabs. You can see him eating away. He's very successful. The blue-leg hermit crabs, and we actually introduced about 22 fish in April. This was really exciting for us. This was the first fish release in the Biosphere 2 ocean since 1996. So it was kind of a proof of concept for us. We were able to show that the system was healthy enough to sustain this kind of life. So we have many more plans for introducing more of these organisms in the new year. But it's no small feat to introduce these organisms. It is not easy. We actually had to pick up these fish at the dock at Tucson Airport, and on the boxes they say live fish, and the guy's like, what are you guys doing? And we're like, oh, we're just putting them up in the ocean in Biosphere 2, and he thought we were crazy. So we brought them back to Biosphere 2, actually swabbed each of them for their microbes, weighed, measured each of them, acclimated them to the ocean water, and about 10 hours later at 12:30 at night, we added them into the ocean. So that was a long day, but very exciting, and they're doing really awesome. So if you come to Biosphere 2, you can actually look in our ocean gallery windows and see these fish eating away. It's pretty cool. So the other thing that we've been doing, this is our algae crew, is hand pulling this algae. So we have been on a mission to do this since June. So we have to date pulled about 5,000 pounds of algae by hand out of this ocean. We will be doing more we plan to pull another 2,000 pounds actually on Thursday this week. So we have been working pretty hard at getting this algae out. And if you haven't been in the last couple months to Biosphere 2, you come and look over it, and there's very visible spots of clear in our ocean, so it's looking really great. Now, the reason that we're doing this all, it's a lot of effort to put in the beginning, but it's all in preparation for bringing corals to the Biosphere 2 ocean. And these are some of the different experiments that our partners are running, and we plan to also include in our experiments out of Biosphere 2. So I will just kind of quickly highlight some of these. Assisted evolution, so this is looking at the corals that are resisting this increased temperature, so the ones that are surviving, and taking those corals and breeding them, creating a stronger second generation of corals. So no genetic modifying, just kind of speeding up what we think will actually happen naturally. The corals just don't have enough time. Probiotics, so this is really neat. There's actually a lab that we have partnered with in Brazil that are working on developing probiotics for corals that make them more resilient to CO2 and temperature. So just like when you have a stomach ache, you take a probiotic to kind of calm things down. It's the same kind of thing. Stress hardening, so this is really putting these corals under fluctuations of stress. So cranking up that temperature, cranking it down, cranking it back up a little bit more, cranking it down. So stressing them out to their tipping point over and over and over again, and they become stronger each time. Like I mentioned, being able to look at the Biosphere 2 ocean at a community level is really important to us. So we can actually bring together animals that we expect to meet each other in the future here as animals shift away from the equator because it's getting too warm near the equator. And we suspect we'll have novel interactions between these animals that we've never seen before. We can actually test that in the Biosphere 2 ocean, introduce animals together that we predict will see each other in the future, but we have no idea how they will interact. And we can also test technology. We can test these things that are being invented. We can test different techniques. It allows us to really have an inside look at how these things affect ecosystems. So this is just a look at some of the probiotic work that we've been doing. We actually have a PhD student that worked with us from Brazil. She's back in Brazil right now, but we got some corals shipped to us here in Arizona from Hawaii, and we're able to really understand their microorganisms. And Erica has been working on identifying those microorganisms to develop the probiotics for them. So we will have these corals back in the Biosphere 2 ocean soon. We've actually been working with the lab in Hawaii to understand some of the corals that we potentially will be putting in our ocean. So coming January, end of January, which is crazy, that's like a month away now, we will actually have corals at the Biosphere 2. So we will have them in those raceway tanks, starting to do some experiments, and with hope of putting corals in the large ocean system this coming year, in 2020. Now I want to highlight some of the things that our partners are doing around the world when it comes to reef restoration. There are a lot of reasons for us to have hope. I wouldn't be standing up here in front of you today if, it was all just never going to happen. Corals are never going to survive. I wouldn't be here. I wouldn't devote my career to this if I didn't think that we had a chance. And our partners and many others are working tirelessly around the world to develop solutions to saving our coral reefs. So Mote Marine Laboratory in Florida has been an essential partner for us and has been doing some really awesome work on the Florida Reef Track. The Florida Reef Track primarily consists of elkhorn corals, and these elkhorn corals have gone through mass extinction. I mean, they have been in really big trouble the last couple of years. And Mote Marine has been able to bring these corals back into the Florida Reef Track successfully. And they're now thinking about how to expand these efforts. About 2 weeks ago, we actually got to visit Mote Marine. So we were in the lab with them. You see me here actually fragmenting a coral. So this is the technique that they've developed that actually increases the growth rate of corals about three times normal. So it's essentially just kind of breaking the coral. I'm using a saw here, but breaking the coral actually kind of triggers a stress response that then allows the corals to grow at a faster rate. So we have our little cut Biosphere 2 corals that we've put in a tank for them to continue to grow. This is the scale of the research that's happening around the world. So this is just kind of an example of this is the top institutions doing reef science, and this is the scale that they're working at. So you can see the need for a system like ours at Biosphere 2 to really expand this research. This is at CSIM in Australia and at Mote Marine in the Florida Keys. There's also work being done on looking at how to restore corals that have been damaged because of storms. So Mars has been working, Mars like the candy that you get, like M&Ms, has a piece of their company that is devoted to coral restoration because most of their revenue actually comes from the pet industry, so cat food, which requires fish. So they're pretty invested in making sure that there's still coral reefs around so that they continue to produce cat food. But they've developed these things called spiders, and they're able to secure coral fragments that have broken off due to storm damage on these spiders to be able to regrow. Now, technology is crazy, as you all know. There's constantly something new on the market. This is also happening in coral reef restoration. So 3D printing has become a conversation piece for a lot of scientists, thinking about how do you use 3D printing to restore corals. The most practical use at this point in time is using it as a plug for these corals, where you secure these coral nubbins on when you're restoring them. But there's also been some work being done on having 3D printed reefs while reefs are undergoing restoration, so that they keep the fish around. There's still habitat for the fish, so that as the corals develop to be healthy again and be able to provide those ecosystem services again, the fish stay in that area. Now, I'm not going to go through this, but I just want to highlight, this is the amount of work that's being done. There's been genetic and reproductive interventions, physiological interventions, population and community interventions, and environmental interventions. This is all stuff happening right now. This charts were just published this year in a report. This is all work that is being done today. So there are people working really hard around the world to understand and to save our coral reefs. So Biosphere 2 comes into this as a place to bring all of this together, to understand what is the best use of our resources, what is the best solution, what are the technologies that we should be using for this, and to be a place for collaboration. That's a thing in science that's so important. So having a community of reef scientists that are using different techniques, bringing them together to see how those things can integrate, and then really piecing apart which are going to be the best. And there's no one answer. There's going to be techniques that are best for Hawaii, and different ones that are best for Florida, and different ones that are best for Australia, because every ecosystem is different. So we're able to really understand that using the system here. And then we're able to stress them out. That's the beauty, is that we can think about what are these future conditions going to be. And we can put these corals under them to really test these solutions. Is it going to work in 20 years? We don't know. Let's test it. Let's look at the predictions of what temperature is going to be in 20 years, 30 years, 50 years, and see if these corals are going to survive. Because if we can't keep them around for that amount of time, it's not worth our efforts. We need to ensure the survivability of these reefs in the future. Because they're important. We need reefs. So 1 billion people depend on reefs for their food source. $36 billion of money is generated, whether it's because of jobs or tourism related to reefs, and because of the natural protection. Now, unfortunately, this is the reality. This is what we're seeing today. Overwhelmingly, this is what more and more reefs are looking like. So we hope that in the future, this is what reefs will look like. There'll be some interventions, there'll be some engineering, there'll be stuff that maybe doesn't look supernatural, but is on the right track to get us back to this. This is the goal. This is what we want. This is what we want to continue to have around the world. And us having this resource at Biosphere 2, 45 minutes away from where we're sitting right now, is invaluable to this work. That's what I got for you. Thank you so much. So I'm happy to answer questions, but I also want to point out that we have a very active social media, so if you are interested in learning more about not only what we're doing in the ocean, but following along with what we're doing at Biosphere 2 in general, it's a great place to find us is on all these social platforms.
Thank you. And those of you who have been here before, you know that this is when we start raising hands and asking questions using the microphone. As I see hands go up, I'll get to people in the order that I see the hands go up, and I'll bring you the microphone so that everybody can hear the question that's being asked. So there's one right up front here.
Katie, thanks for a great talk. I'm just curious, which part of this research are you going to turn into your PhD thesis?
That's a great question. I do not know yet.
What part of it is?
Potentially. I haven't gotten there yet, but so I have my bachelor's in science, but I got this. I actually interned at Biosphere 2 when I was an undergrad at the university, and was offered a full-time position when I graduated. So I've been out at Biosphere 2 for about 7 years now. And yes, I think in the future a PhD will come out of this, I hope. But there's lots of interesting work, and the folks that we get to interact with are nothing but inspiring.
Thank you. There was a hand over here.
Thank you for your presentation. So I have two things to ask you. One is, do you have this presentation in some form of social media and a video that can be sent to somebody who lives outside the United States?
I don't, but I would be happy to share it with you. It's pretty big, so I can give you one of my cards, and I'd be happy to email it to you.
Okay. The second one is, and it would be this person that I send it to. It's a water engineer who works in Bahamas. He works typically on commercial development, which means the creation of villas and yachts and marines. And when those are created, they have to mitigate by restoring any marine life that's disturbed. And sometimes they want to restore marine life even when they haven't disturbed it, because they have this small problem with a thing called hurricanes. And the question is whether the use of rock weirs to create reefs and to try to generate corals is proving to be effective or not. In Florida off the coast this has been used, and in Bahamas they may be trying to introduce this, and basically to just slow down the water waves. But the research I read showed that one subspecies of species of fish was able to adapt to that, and another one wasn't. So any information or opinions you have about that would be appreciated.
Yeah, I mean, I don't have a lot of specifics to share on that, but I know there are a lot of interventions being developed right now related to that, and we don't know much yet. I think there's a lot of research to be had. There has been cases of putting these things in place, not fully understanding what those implications are, So not knowing what those implications are until you start seeing consequences in the natural systems. I don't know specifically on that example what we've seen so far, but thinking of a couple others that we've seen, where they've dumped concrete and around reefs to try to develop reefs, and everything around it is like, what the heck is this? This is nothing. I can't build anything on this. So there's lots of things that are being tried, and I think that where Biosphere 2 comes into this picture is that we can actually try those things out here and see how that affects the natural system, how it affects the system as a whole, so that we can make more educated decisions of putting these interventions out into the natural systems.
All right, I see another hand go up right here. Here comes the microphone behind you.
The Great Barrier Reef seems to get a lot of attention in the press and otherwise. Is it deteriorating faster than other reefs around the world?
So it's a great question. I do not think that the Great Barrier Reef is deteriorating any quicker, but it's so much larger that its impact is greater. So the amount of surface area that it covers, the loss is larger. So it's deteriorating at the same rate as other reefs around the world, but we're seeing greater impact in the Great Barrier Reef because of its size.
Thank you. Oh, I see your hand over there, but I have another question over here first, so I'll get to you soon.
I think that ultimately the health of the coral and the reefs have to do with the environment that they're in. So you've talked about heat pollution or increases in heat, but there are also increases in acidification, and there's nuclear pollution, and then there's all sorts of other chemicals I've never even begun to dream of. Those things are going to have some sort of solution before your program is going to be able to work. Do you understand that? And have you given thought to that?
Yeah, absolutely. So the reason that we focus on temperature is because it's a global issue, and it really, truly is the #1 threat to corals today. Now, not to say that they're not undergoing many other threats at the same time, and that's very true, and we see that with nuclear pollution, with runoff, with agricultural runoff, we see different nutrients. It's hard to pick one thing, right, to focus on, and you really can't. So we plan on running dual experiments once we kind of understand this temperature thing, adding CO2 to the mix, understanding how the acidification and temperature, because likely they're being hit by both, affect that. And then maybe in some of the raceways, we're able to increase maybe some of the nutrients that you might see from runoff. And the nuclear areas, the big issue in nuclear areas is temperature, more than chemicals, in a lot of studies that I've seen. So, yes, I mean, there's a whole list of issues that these corals are under. We just have to kind of pick and choose of what we're focusing on at this point in time.
Thanks. We've got another question right here.
Two very unrelated questions.
No problem.
Okay. One is, are there any areas in the world where reefs are doing okay, or where they're showing survival stuff? And the other one is, what do you do with the algae? Can the algae be used for stuff?
Great questions. Yes, there are areas in the world that the corals are doing okay. We've seen parts of the Red Sea be able to really withstand these changes a lot better than some of the other places, which has been really interesting to see. We have some areas that are way more protected, or they have natural upwelling that hasn't been affected by temperature yet. So there are some places, they are far and few between at this point, unfortunately, and they are constantly changing. We'll get new information every week that says, oh, now this place is undergoing a bleaching event. So we're seeing less and less of that, unfortunately. But there are places, like anything on this planet, there are some species that are resilient and able to survive these changes, depending on the ecosystem that they are in. In terms of the algae, it's a great question. We have really thought about this and tried to find a use for the algae. We've contacted people at U of A that are doing, biofuel We've looked at composting it. We've looked at giving it to a farm. And it is just too dang salty. We can't do anything with it. We've tried certain things, but we can't put it on the ground due to regulations because of the salt. So we can't, compost it. So we actually throw it away. It's the only thing that we've been able to do with it. Unfortunately, if anyone has a better idea of what to do with it, is gross, by the way. So don't raise your hand too quickly after it has a few days out of the water. You don't want to be around us when we're lifting that stuff out.
Thanks. We got another one right up here.
Yeah.
Thanks for the great talk, Katie. Are you using shellfish like oysters to reduce the algal load?
That is an awesome question. Yes, we do have some oysters in the system. So oysters are awesome filter feeders. We also have a few sponges in the system, so we will be continuing to add those organisms as we go further down this road. What's kind of inhibited us from adding those organisms is we just have too much algae. There's a surface area. So as we've been able to take this algae out, there's more space for these animals to be able to inhabit and to be able to have a reasonable amount to consume. What we put the fish up to in the beginning was a big task, and we quickly realized that we need to intervene. Shellfish will play in our system.
Oh, sorry guys. Coming back again.
As the
Talking our aquariums. I think that the corals, there's more and more being developed in terms of being able to breed them in labs. So being able to have what Marine has been able to do to fragment these corals, so more and more corals are being grown in laboratories, which are, this is optimistic of me, but I'm hoping that it will become more and more of what we see in the aquariums is lab corals versus ones that are being harvested out of the ocean. But it has immense impact. tissue. Work being done on developing the stronger zoos of belly that can live in these corals as well.
So once that material is expelled, what happens to it at that point? Is it sort of live bar involved?
Yeah, it's in the water column, but most of the time it probably dies off.
Okay. My curiosity, as the oceans warm up, perhaps the fish can move a bit, so I was wondering how that affects their metabolism. When they're in the deeper cold waters, you have different strategies for keeping the heart, the brain, the eyes, or flippers.
So in terms of metabolism, I'm not 100% sure about that, but because they're able to move away from it, they also are not as sensitive to temperature as corals are, but we see an increase in disease when temperature goes up. So we see the fish and other invertebrates actually being more affected by that than anything else.
So would that be a way of communicating disease with predators?
Oxygen. What effect does this have on corals?
That's a great question. Well, so coral, that ocean chemistry, so when you lose that oxygen in the ocean, you're also, losing those ions that are really dependent on it. So I think it would also affect the coral being able to build a proper shell. I think most of all, it's going to affect the organisms that are around the coral to maintain the algae not growing on top of it. If the fish aren't...
It's affecting the fish.
Yeah, absolutely. So it's definitely affecting the fish, which fish are necessary in reefs to keep the algae at bay, away from the corals. So I think that would probably be the biggest effect is those other animals that the corals rely on to keep them healthy.
Katie, I'm curious, is the algae that lives in the coral species specific?
It is.
So that it's super complicated.
Very complicated.
Yay! And then, if you could pick 2 places in the world to go and scuba dive, where would they be?
Great. Well, so I am actually heading to the Great Barrier Reef in February. So my cousin's deployed out there right now with the Navy, so I am going to stay there for free and dive the Great Barrier Reef, and that's definitely a bucket list for me. And because I'm a scientist and a little twisted, I want to go out and see all of these reefs that are actually dying, being degraded, so that we can fully understand. And there's been a mass bleaching event off the coast of Hawaii this past couple months, and I'd actually would love to go out and see what that looks like. I have not witnessed that myself happen, so I would enjoy that. Well, kind of, not really, but yeah, like I said, twisted.
Thanks a lot for your presentation. Really appreciate it. I have a question a little closer to home, the Sea of Cortez, and I wonder if you have some projects which relate with the Mexican folks.
Yeah, we do. We have a couple of our partners that are down off the coast of Cancun that have some coral species living out there that we have started to work with their lab on a few projects, but not extensively yet. Getting permits to get corals from Mexico is very complex. So we are starting with the states because we can get permits pretty easily from Hawaii and Florida to get these lab-bred corals. But to expand outside the states becomes a very big challenge for us to be able to source these corals. So we hope to use the corals that we can get permits for to then inform them on techniques that are being successful that they then can work in their laboratories on. But down the road, we could get permits. I don't know. So far, not looking great.
Another one here.
I'm curious, when you import corals, what volumes are you talking about?
So initially, for the first round in January, we're talking about 250 fragments. So these are these nubbins that are, on the sides, and then maybe a few that are about this size, so maybe 10. So small amounts to start. But at Moat Marine, we've talked about getting about 150 dinner plate sized corals. So just depends as we understand what is available to us and what we can get permits for. But starting decently on the small side, we want to get our feet wet before we dive in too much here. Well, I have a lot of ocean puns right there. I should.
I don't see any other hands right now, so I get to ask a few questions.
Yeah.
So I know, if I understand correctly, a lot of these bleaching events are related to warm waters that come in and cover the corals for a period of time, and how. So overall ocean temperature is warming, but of course there's warmer currents and cooler currents. Can you talk a little bit about how temperature moves, circulates within the ocean, and is it circulating all over? Yes, okay. And then does it relate at all to depth? So most corals are growing in relatively shallow water. I'm just wondering, how does that warmth move, if you can address that?
Yeah, so we really see major changes in that during El Nino and La Nina years. That's when we see these major fluctuations. So during El Nino years, we see upwelling, warm water coming up into these reef areas. So that is kind of a major point of when we see the warm waters moving up into these reefs. But I mean, the overall ocean temperatures are warmer. There are upwelling events that occur that are warmer. There are ones that are cooler. It's the oceans are moving. So I don't know the full dynamics to be able to lay out to you right now. If we got a whiteboard together, maybe later we could go through it. But yes, there are a lot of dynamics at play, and I would say the most significant effects that we're really seeing is during those El Nino and La Nina years.
And then in the presentation last month, as part of the same series, we were learning about the fisheries off the coast of California and the current that comes through there. And one of the issues, again, for the fisheries was ocean warming and how that affected different species of fish. When these warmer waters are sweeping over these coral reefs, the coral can't move, but the fish could swim away. Do the fish suddenly leave when that happens, or do they hang around the reef?
We see both occur. It really depends on the area. It depends on where the next reef is in proximity to the reef that you're looking at. There's not one answer to that. It is totally different in every place that you go. It depends on the species of reef fish that you're looking at. So there's not one answer to that. But yes, we see differences. We see some fish migrate off when the water gets warmer. We see others that stick around. Just depends.
I get you. Oh, here's another question.
So if college students want to volunteer or intern, how would they go about that process?
Contact me. Yeah. So I've got my card up here. Feel free to grab one. My information's all over our website as well. So we try to engage as many interns as we can handle as a team. And we're a small team, so The more hands, the better.
And if anybody else has a question, I'm going to ask one more question.
Okay.
Which was, something I had never heard about before was increasing the amount of light that reaches the Biosphere 2 ocean, which has never been done before. Is that because most coral reefs would be exposed to more light than we receive at this latitude?
It's more due to the big glass windows above the ocean. So it blocks too much of the necessary light that these corals would need. Primarily, the main reason is that we would like to put corals at depth as well, utilize not just the reef crest, but really the full system. And the ocean does not get enough light. Those windows really block it. So that has never been done before, true, but it's necessary to be done for what we're looking at.
Interesting. Here's another one.
Hi there, wonderful speaking. I was wondering, what sort of lessons have you learned both at Biosphere and in your own research which might be applied to future human habitations on the moon, Mars, and other worlds?
The Earth is complicated. It's super complicated. So I think that the thing that I have really, as we've started to monitor the system, and me being able to be there for an extensive amount of time, spending 7 years working on the system, and still going in and finding things I had no idea were there, just constantly amazes me, and I think proves the fact that as we consider habitating other planets, one, it's essential for us to understand this one first. If we don't fully understand what it takes to sustain life on this planet, we're not going to be able to do it on others. We haven't quite gotten there, I don't think. So I think just kind of really making sure that we're doing our due diligence to understand what's going on here before we venture off. So really just to continue to be open-minded and not think that you know everything. There's always surprises, which is something that I've really experienced out at Biosphere 2, which has made my job really fun.
Thank you. I have one last question, if nobody else does. I don't see any other hands going up. So again, at least in the Biosphere 2 ocean, corals died and algae moved in.
Yep.
So part of my question is that also the same cycle that happens if a coral reef bleaches? We always see these images of these white corals. At a certain point after the corals have died, does the algae move in and then cover those corals?
Yes.
And if that happens, are there any natural processes that then might remove that algae or eat that algae? Or what happens to that whole ecosystem if suddenly all that algae moves in?
Yeah, so yes, it is what happens. So when we see the bleaching of the coral is not yet dead. It has a chance to recover, but if it does end up dying, algae takes over rapidly. It's actually how you can, when you're on a reef, you can tell if you're looking at a live coral, a bleached coral, and a dead coral, you know the dead coral is dead because it has algae filaments coming off of it. It's pretty obvious.
So I'm curious to interject here. The bleached coral is not yet dead.
It's not
How
dead.
does the bleached coral keep the algae at bay? Like, why doesn't the algae move in on the bleached coral?
There's a couple dynamics at play there, but I think the fish are still kind of keeping it away. There's still some hope. They're still filter feeding, so they're still active. Those polyps that live in these little holes on these corals, these little pores, are still active. So there's movement happening on the organism that keeps the algae away. So it's a pretty true state of death when you see algae overgrowing a coral.
And then are there any natural processes after the algae has moved in that would reduce the amount of algae, or does the ecosystem just shift to like it's all algae all the time and nothing else can live there, or what happens after that?
So we have seen a couple examples of this happen. One that I can think of that's a great example is off the coast of Coconut Island in Hawaii. They saw this occur. They had a mass bleaching event in 2016, killed about 90% of its corals, and they saw algae really move in. And there was no natural intervention that was occurring. So the science team over on Coconut Island actually introduced urchins to eat the algae and developed something called a super sucker, which I'm super jealous of because it's a boat that has this essential vacuum on it that you vacuum up the algae and then drain the water back into the sea. So there's been some human intervention, and they restored that reef, by the way. It was successful. That's obviously small scale. It was a small area. Doing that on a full-scale reef would be almost impossible. So that is why we are trying to understand what a degraded system looks like and what some of the interventions might be able to be to go back to a healthy reef. I'm sure nature will play its course, and if we give it enough time, something will develop and evolve in those areas. But we're going to try to prevent that happening. We're going to try to prevent those reefs from getting that degraded.
All right. Well, thank you. That was fascinating and exciting to learn what's going on right here in Arizona at Biosphere 2. Some of you, we now have feedback forms at every single one of our science cafes. So if anybody would like to fill out a feedback form, please do. I've got pencils up here and feedback forms. And I'm excited to announce that our Spring 2020 Science Cafe series will be called From Science to Stewardship, Earth Science in Action. So it'll be a series of presentations from researchers who are translating their research, which is all earth science research, into programs and decision making that helps us maintain healthy landscapes and manage our lands in a sustainable way. So I hope you'll stay tuned for that series in the spring and come join us again. Thank you so much, Katie, for a wonderful final cafe for this Ocean Science series, and happy holidays to everyone. I hope you have a good winter break.
Shipper, what is the science lecture series on? They announced that yet? I haven't announced it yet.
I know this, but it's.
Should I look it up?
It's slightly abstract. Katie, do you remember?
I know it, but I can't remember it either.
It's kind of abstract. That's the thing.
And it's 4 lectures, right?
It is 4 lectures. Darn.
Catalysts of change. Catalysts of change.
Catalysts of change. What does that mean? I'm not sure.
Life beyond Earth, our rapidly changing biosphere, the promise of peril of artificial intelligence. Our world is changing faster than we are.
Yes, and we will not overlap with the lecture series this year. So in February, when the lecture series is happening again this year on Tuesdays, they used to happen on Mondays, but it will be happening on Tuesdays. In February, this cafe will be on a Thursday. So check out the calendar, mark the dates, and we look forward to seeing you in January. Happy holidays.
What? Yeah, what?
Oh no.
My wife gets it, and I all
So if you go to the Science Cafe site and submit your email, then you'll get the email saying, is this you, or do you really want to sign up? And you say yes, and you should be back on there.
Okay, but somehow...
I'm sorry to hear that. I hope that's not happening often.
Well, I don't know what happened is the thing. Because, I mean, I had a computer that totally crap out on me once.
I've had strange things, but my wife is getting it. Matter of fact, I was going to look on the website for tonight, and she told me she just got the email.
Right. Oh, good. Well, I'm glad she's still getting it. That is really confusing.
Okay, I'll try and do